What's up, guys? Welcome back to another lab. Today we are going to be doing the reproductive system, and let's check it out. We are going to go through. Tell this one's a little bit longer. We're going to go through the male and female reproductive system. For each of those systems, we will name structures. We will see some histology slides. And I'll talk a little bit about kind of the lecture material, but nothing super crazy. As far as spermatogenesis and oogenesis. And then at the end, you will see, obviously, some real anatomy pictures and a few conditions associated with reproductive disorders. These are the terms you need to know to go through, kind of check them off as you're studying. Let's do it. Starting off with the male reproductive system. Obviously, this is a sagittal cross section. You can see the cartoony picture from your textbook and the cadaver picture at the bottom. I will highlight the important structures and then I will show you where they are, as always, on the models. First thing we got is the penis, right? We will see, I guess, models mainly, a couple cadaver pictures. That is the primary defining structure of the male reproductive system. We will go to where sperm is produced. Obviously, the main goal of this is sexual reproduction. Sperm is produced in the testes. We will take a look at some of the substructures within the testes. The testes are held by the scrotum. This is very important. There are muscles that can elevate and descend the testes in order to regulate temperature to help the sperm not die, right? After the sperm is produced in the testes, they are stored in the epididymis, and when necessary, they are released and come out through the vas deferens. Here you can see the ductus deferens. I don't care which term you use. I always use vas deferens. And that is the structure that will carry, that's not the color I wanted. That is the structure that will carry the sperm from the epididymis up, out, and around, passing through the seminal vesicles. Shouldn't say through, by the seminal vesicles, which will contribute some contributions, fluids, things like that, to the semen. And then it will come out through, where's my term? The urethra. In the male, the urethra serves two functions. One, obviously, semen, ejaculation, and two, urine. So in addition to those reproductive structures we will see, you will also see, this is called the ureter. That connects to your kidneys, and that is where the urine drains down after it is produced, and it will be stored in the urinary bladder. These are your major structures. Let's take a look at where these are. I'll circle the seminal vesicles. I forgot one. The prostate. The prostate is here as well, sitting inferior to the bladder. I will highlight it for you right there. And we'll talk about the role of the prostate, mainly in lecture, as far as contributions to semen, etc. So what can we see on here? You'll notice that there's two pictures. There's one model and there's another model. So I'm just flipping back and forth between the slides. The main difference is this cover, right? On the second model, the cover is removed. For the male, there's not much difference in what you can see. I will highlight some of the structures we can see here. I think you can see them a little bit better on this one, but obviously I'll label them on both. Let's start off with the order we talked about before. testes, scrotum, epididymis, right? Vas deferens comes up. It's going to come around, down, and out. So clearly we have the penis and the urethra as well. You can see some accessory structures here, like the prostate and the seminal vesicles, as well as the urinary bladder, right? The last one is the ureter. Which was not labeled on the other one, but I drew it in for you, drew an arrow. And that again takes the urine from the kidneys and drains it down and fills up the bladder. And then when your bladder stretches, you're ready to void your urine, and it comes out the urethra. Pretty straightforward. We can see the same structures on here, right? Testes, scrotum, epididymis. The vas deferens is cut off here. Obviously, it would run up and around. So not the better one to see is probably the other picture for the vas deferens. We can see the bladder. We can see the prostate. The seminal vesicle is kind of right there. It was better to see on the other one. We can see the ureter, obviously penis and urethra, respectively. Pretty straightforward. This is the anatomy of sperm. And sperm have a couple different important structures that allow them to do what they do well. Number one, it has a tail. This is called the flagellum. It basically spins and it allows that sperm motility. So that is how the sperm will get from, maybe the entrance of the cervix, through the uterus, up through the fallopian tubes, and hopefully, or maybe not hopefully, contact an egg for fertilization. We have a midpiece, which is this region right here, filled with tons of mitochondria. Swimming is a very energy-intense, laborious process. So we need to fuel it with tons of ATP and energy. And that's the role of the midpiece. The head, unfortunate. The head contains a few important things. It contains the acrosome and the nucleus. The acrosome is this little capsule kind of on the head of the sperm that contains enzymes that are very, very important when that sperm does contact the egg to allow it to dig through, as well as the nucleus, which is obviously really important because it contains the 23 chromosomes responsible for carrying the genetic material from the male to the female egg. The female egg obviously will have 23 as well. When you have 23 and 23, they fuse together, you will get 46. And I'll probably draw that picture for you in a little bit. This is called a gamete. Gametes are sex cells. They are very different than all the other body cells in your body. And that is because they are haploid, or they have half the number of chromosomes responsible to form a full diploid cell. So that is the basic parts of the sperm, and we can see them here, right? We can see the acrosome, the nucleus in that head. We can see the midpiece, and obviously we can see that flagellum. You can also label structures on this picture as far as the male anatomy that we talked about previously. And now, to kind of almost wrap up the male anatomy, obviously we have some histology and things. This is a process called spermatogenesis. It is important. We will talk a lot about this in lecture. But sperm production, basically what you have is you have one cell that turns to another cell, that turns to other cells, that turns to other cells. But the key factor here is that you end up with functional sperm, right? So I kind of simplified this picture, if you will. from what you're looking at here. And the keywords, try not to close this thing out here. Spermatogonium. That would be this first cell. The spermatogonium are the cells that we always keep kind of, and we'll look at the testes, towards the outside of these seminiferous tubules, and then they will start to undergo mitosis and meiosis, 1 and 2. And during mitosis is just cell division, so we can keep plenty of spermatogonium up there. Meiosis has a very important component of crossing over, where basically we are switching up the genetics that will be in these sperm. If you have offspring, none of those offspring are genetically identical unless they are identical twins. So the genetic variability in offspring is created during that meiotic process from the spermatogonium to the primary spermatocyte to the secondary spermatocyte. Then we get spermatids. And then finally, through a process of spermiogenesis, we make spermatozoa, which you can just call sperm. Their actual name is spermatozoa. That's fine. They get stored and released when necessary. So as stated before, right? Spermatogonium, primary spermatocytes, secondary spermatids, and functional sperm. The key thing with these sperms is that they are haploid. They have 23 chromosomes. They are genetically slightly different from each other, and we make a lot of them. So that is the process of spermatogenesis. Obviously, in lecture, we talk more details about meiosis and things like that. This is another image where we're taking a look at it. The key thing here that you will see is that it happens in the testes. So in the testes, we have these little squiggly tubes called seminiferous tubules. We can see that term here. These seminiferous tubules are tubes, and the sperm production starts from the outside and works itself inwards. So that process that we talked about starts from the outside and spermatogonium to the primary spermatocytes to the secondary to the spermatids, and then on the inside of that tube you will have functional sperm that will then be shipped out, right? So keyword here, seminiferous tubule. And obviously all of the production of sperm that we discussed, ending up with 1N or haploid gametes. Very important. This just highlights the same thing again for you. And now we'll look at some histology. So this is the same picture we saw the testy. You can see inside the testy, we have all these seminiferous tubules. You can see it right here in the gross anatomy picture. And then once that sperm's produced, it'll come out into the epididymis. So we'll just take a couple looks at some histology for the male. This is the testy. Recognize it. What are the squiggly things inside? Seminiferous tubules. This is a pretty zoomed in picture. And on the next one, we're going to zoom in on even more some of those seminiferous tubules. And they look like this. So there are a couple terms I have to add in here. One of them will be interstitial cells or Leydig cells, right? Those Leydig cells can be seen on the outside or in between those seminiferous tubules. The testes not only produce sperm, but as we talked about in the previous lecture, the testes also produce hormones. Testosterone is produced by these Leydig cells or interstitial cells, and that's why they're there. As we just discussed, the sperm production starts off towards the outside and works inwards. And then your sperms will be in here. Probably not the best color, but it's all good. So those are obviously the yellow thing are the seminiferous tubules, seminiferous tubules, seminiferous tubules, and the cells outside of those in between are Leydig or interstitial cells. This is a slide of the epididymis. The only thing I really want you to know about the epididymis is that there's sperm in here, right? We could see spermatozoa in the lumen. There are these little cilia-like projections in the epididymis, which helps us kind of filter out the sperm and move things along. And you will also see during times of ejaculation the requirement for some smooth muscle. And that smooth muscle is the dark stuff located on the side. And that's how we move things along, right? So this slide, epididymis. This slide, vas deferens, right? There are a couple different layers of, might ask you. But clearly inside the vas deferens, there are sperm, right? You have the innermost layer, which is the epithelial layer, and you can see that here is kind of highlighted in the darker color red. You have muscle, obviously, because the vas deferens have to move this sperm through, and that muscle will be this layer here, all that darker stuff, usually muscle. And then the serosa is just the outside layer. You don't have to stress about that, but that would be that lighter color towards the outside. So again, slide is the vas deferens, and we have 3 different layers of it, as well as sperm in the middle. This one's easy. Seminal vesicles, recognize it, right? Prostate, recognize it, right? It has these big kind of sacs, if you will, or spaces. These are called alveoli, but whatever. And you're going to have some prostatic fluid inside there. And now we're going to look at the penis. So 3-ish major parts. Clearly you could see it up here. You can also see it down here. This is a cross section. The first, you can see this tissue is kind of very filled with a lot of space. This is kind of what engorges with blood during an erection. And you can see the corpora cavernosum. Those are those big cavern-like structures that will fill up. Down here, you can see some soft tissue around the urethra. And the urethra, I'm going to highlight in red here, that is the opening. And the tissue surrounding it is called the corpus spongiosum. So kind of tricky, new words, maybe if you didn't have it in lecture yet. You have the corpora cavernosum and the corpus spongiosum, and then you have the urethra. And obviously we talked about the function of the urethra before, as far as transferring semen as well as urine. Check. Here we are looking at it. In this situation, it is not engorged with blood and stuff, so our corpus cavernosum is located here. The corpus spongiosum is located here. And the urethra is located here. And that's what the histology slide of the penis looks like. This is just a higher resolution picture of that seminiferous tubule, right? So we could see that sperm production happening, that spermatogenesis inwards towards the middle, and then in here we will have our functional sperm. Seminiferous tubule, obviously found in the testes. This is the same thing, zoomed in a little bit more, but we can see one new cell, and that new cell is here. And it is called, I knew that was going to happen. How did I know that? I don't know. Sertoli cell. So that Sertoli cell is, we call it kind of like a nurse cell. It helps, you can see this sperm production occurring here, and then obviously sperm in the middle. It helps facilitate this process, nutrients, etc., and Sertoli cells are scattered throughout that seminiferous tubule to aid in that process. This is a picture of sperm. Obviously, we could recognize the head, the midpiece, and the tail, respectively, also known as the flagellum. And we made it through the male. The male's pretty straightforward. Recognize your anatomy, couple histology slides, you're good. The female is a little bit more complicated. We'll try to keep it relatively simple. There's a lot more going on. We have to talk about hormones. We have to talk about ovulation and things like that. So it's a little bit trickier than just we start making sperm from puberty till forever. We got eggs, right? Oocytes, oogenesis, some anatomy, as well as some histology. So let's take a look at some of the structures of the female reproductive system. Need that, so I'm trying to move that out of the way. First off, let's start where everything begins, and that is with the ovary. The ovary, which red is going to be a bad color, is that little structure in there. We have two. And the ovary is where the egg development is taking place, the ovulation takes place. Once that egg ovulates out, once per month, I won't even do that. It travels down the fallopian tube. The fallopian tube is also known as the uterine tube. Either term is fine for me. And it will travel down out here, down that fallopian tube, into the uterus. The uterus is important. This is where we have multiple layers, right? Specifically, the endometrium grows and sheds every month. This is where, if fertilization took place, implantation would happen. This is where the developing embryo would divide and turn into a baby, and it would live for 40 weeks until it comes out down here. So we have uterus, we have the cervix, which is the entrance or exit between the uterus and the vagina. Obviously, we have the vagina, both involved in birthing a child, as well as sexual reproduction, because that is how you would get the sperm into the uterus and fallopian tubes, respectively. And we see some structures towards the outside, covering the female reproductive system and urinary system, called the labia majora and labia minora. You can call them the labia majus or minus. And we can see we have the clitoris here. And there are some urinary structures. The first one being the urinary bladder and the urethra, where the male had a double function urethra that was longer and carried semen and carried urine. The female reproduction is separated. So you're going to have the reproductive system independent of the urinary. Obviously, the bladder, we will probably see on the model. We can also still see that ureter that drains urine into the bladder. So that bladder, and then you can see the urethra is much shorter in the female because the bladder's right there, right? It serves one purpose. So pretty straightforward. This is a picture to specifically highlight the reproductive components. So on here, we can see the uterine tube, right, which would be this, and the ovaries. Now, this is an important picture that we will take a look at in a few minutes. Right here. This is what's happening in the ovary. Now, we'll go into it in lots of detail in lecture. But for the most part, you have the maturation of the egg develops, it turns into something, it grows more cells, then that egg is ovulated. Some of those cells stick around just in case fertilization happens, and then they will atrophy. So that is where it's happening, and then it switches to the other side, and then it switches back. And once a month, this developmental process of the egg or oocyte occurs, and then will be released. You can also see on here the uterus, the cervix, aka cervical canal, the vagina, and an area that we're going to get to in a little bit are the 3 layers of the uterus. I like this picture because it's pretty simple. The endometrium is the inner layer. The myometrium is the middle layer, primarily made of very strong muscle. And the perimetrium is the outermost layer. Other than that, I think we pretty much covered all this. And we're good. So let's label it. Just like we saw with the male, you have the model with the cover on, and then we're going to take the cover off. In this situation, I prefer the cover off because it's a little bit easier to see these structures. So let's highlight what we know. Number 1, this is the ovary. Number 2, this is the fallopian tube. This is the uterus. We can see here that cervix, right? Uterus, fallopian tube, and ovary. And then we have the vagina, labia majora and minora and clitoris, respectively. And then our urinary is going to be the urinary bladder and the urethra. The only other thing I'm going to add on here is the ureter. Do not confuse urethra with ureter. And we can see the round ligament. I'll probably talk about that a little bit more in lecture. But that round ligament is located right here. And it's responsible for kind of suspending that uterus and stuff. You don't have to stress about that now, but that's where it's located. We can see the same things on the gross anatomy picture. And now we'll talk about oogenesis. So we discussed the sperm, spermatogenesis, and the production of it. The oocyte is similar, but there are definitely some differences. Number 1, we make 4 sperm per meiotic division. For the oocyte, we only want 1 egg. And we'll discuss that. So we start off here with some primary cells. They're called primary follicles. A female has all these eggs basically forever. I shouldn't say that. She has them to start off. And then as they grow, ovulate, released, eventually the numbers will deplete. And you will get to a point where you basically don't have any more eggs left. And the menstrual cycle stops. Call that menopause. And clearly we'll discuss that a little bit more in lecture. So the primary follicles start, we basically select one, and now we have our oocyte. The oocyte is what will be the egg that will be ovulated. As it continues through this process, it will get more cells around it. So you could see here that that's the main egg, and then it starts to grow more cells. And as it starts to grow more cells, we call the whole structure a follicle, right? So we have kind of, we'll call this a primary follicle here, but it's not labeled. And then you have a secondary follicle. So as it continues to grow throughout the month. And then what you will have is you will have a mature follicle. And the mature follicle is easily recognized. You can see the antrum, which is this big fluid-filled space. You can see that oocyte in it. There is something called the zona pellucida that we will probably talk about more in lecture. And then it is ovulated. So I call this, all those cells around, I call it its friends. And then it gets ovulated and it keeps some of those cells, right? Obviously, it gets ovulated into the fallopian tube, comes down, possibly fertilized, possibly not. If not, you get rid of the egg and all that endometrium and reset for next month. If it does get fertilized, that's a different story. So once that is released, right, you have all these little cells that hang out and turn into a structure called the corpus luteum, right? That is what it looks like. And it's just those remaining cells. They turn into an endocrine gland. If fertilization occurs, they release certain important chemicals that we will discuss. And once fertilization does not occur, that structure turns into a corpus albicans, and the process basically stops, and then it gets reset in 2 months or a month after that month. Obviously, you have two ovaries, and they're alternating. So that is kind of the maturation, if you will, of the development of that oocyte upon ovulation and after. It's an ovary. The thing I don't like about this picture is it doesn't show the things in order, but I will show those structures that we just discussed. So right off the bat, these are the primordial follicles, right? They're just hanging out there waiting to go through their developmental process. Obviously, this little thing in here is the egg, the oocyte. This is our primary follicle, secondary follicle, mature follicle, and that's where that egg is being ovulated. So you can see that oocyte gets released from there, obviously exiting the ovary. What remains will be the corpus luteum, eventually turning into the corpus albicans. So the tricky thing about this is it doesn't go in exact order, but you should be able to recognize, a follicle or corpus luteum, maybe ovulation, mature follicle. So the process of oogenesis is a little bit different than we drew with the sperm. Keep in mind, right, if you have an oogonium, which is your first cell, it obviously does mitosis, and that's how we get all these, primary oocytes and things like that. But as we go through the process of cell division, and I'll just simplify this for you, we really only want one ovum, one secondary oocyte. So there's a slightly different process that occurs, and this thing is actually here. All of the things that get destroyed are called polar bodies. So we still have an oogonium, we still have primary oocytes, we still have a secondary oocyte, but the key thing is that secondary oocyte still has two sets of genetics, if you will, and it doesn't complete its meiotic process unless it gets fertilized. You can see here with fertilization, when it does get fertilized, it gets rid of that second polar body. Neither here nor there. The key thing is that egg should have 23 chromosomes when it fuses with the 23 chromosomes of the sperm. 23 plus 23 equals 46, and this is a gamete. But the egg that is ovulated is called a secondary oocyte, right? So you can see a couple key things here. Obviously, we'll talk about it more in lecture as far as fertilization. And that's your oogenesis. Highlights it here as well, talking about the follicles and the follicular stages. And this picture, obviously, we're going to talk about in lecture. This is showing you what's happening during this almost month-long menstrual phase from different aspects. The top part is showing you the follicular and luteal phase. The follicular is when that follicle is developing. The luteal phase is when you have your corpus luteum. But it's showing you the hormones involved, right? This will become important when we start to understand how this is regulated or how it's manipulated through various drugs and things like that. So here we have the ovarian cycle, and it's just showing you as you go along that process I already discussed. Primordial follicles, primary follicles, secondary follicles, mature follicles, ovulation, corpus luteum, corpus albicans. Down here, it's showing you the uterine phase, aka what happens to the endometrium, right? So that endometrium will thicken through that follicular phase, right? That we call it the proliferative phase. It will thicken. It's preparing for pregnancy, basically. You can see it goes from here to here to here. It's preparing for implantation. If fertilization does not occur, right? Obviously, that secretory phase is when it gets pretty thick, proliferative, secretory. And then you have the menstrual phase, which basically we reset. We clear out that endometrium so it can be prepared again for the next month for potential fertilization. But obviously, that's the period. A little bit of histology. Not too much, nothing super crazy. But we can see here the difference between, obviously, this is the ovary, the oocyte, and the secondary follicle. The secondary follicle is the whole thing. Remember, the oocyte is the egg, and then the follicle is the egg with all of its friends around it. So in this situation, we can see that oocyte as well as the follicle. Here, we are looking at a mature follicle. Obviously, in lecture, we're probably going to go through all the specific parts. Key factor here, that is your oocyte, right? You can see the antrum and things. You don't have to be able to label this in crazy detail for lab. Obviously, in lecture, we're going to go through everything that happens with the cells around that egg, the antrum. But for now, this is a mature follicle, and you should be able to recognize the secondary oocyte inside there. Just showing you the same thing, slightly different picture, right? Where is it? Secondary follicle located. Well, it's just showing you that's what it is. And then our primary oocyte is on the inside. So this is not mature, right? This is a secondary follicle. Corpus luteum, big, fluffy, cloud-looking structure. Just recognize it. This is a picture of the fallopian tube. So what it looks like on the inside of the fallopian tube. It's got these fimbriae. It kind of helps the egg move along. It's important. And now we'll look at the three layers of the uterus. So obviously we have the start off down here. Boom, boom, boom. The perimetrium's on the outside. The myometrium's that thick muscle on the middle. And then obviously the endometrium's that thing that we just talked about that gets thicker, 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 and then will be removed. When it gets removed, it comes out the cervix and the vagina. Makes sense. This is just showing you uterus, right? Endometrium, myometrium, perimetrium is always that little guy out there. And here's some histology slides. No, this is the uterus, and this is the proliferative stage. This is when it's, thinner, if you will. You can see here the difference between the endometrium and the myometrium. And then that uterus is not very thick during that proliferative phase. Obviously, it will start to thicken during the secretory phase. This is all endometrium. We can't even see any myometrium on here. You can see that it gets kind of glandular, starts stretching out, getting bigger. And then the menstrual phase is when we are getting rid of that endometrium, right? So you can see a slight differentiation between that myometrium and the endometrium, aka recognize the slide. This is a picture of the cervix, entrance or exit to the uterus. This is a picture of the vagina, has its specialized epithelium that we will discuss probably in lecture. But this is the slide of the vagina. And we have I think we have one slide on the mammary gland, right? So in here, and we'll talk a little bit about pregnancy and lactation, right? You have these alveoli, these little milk-producing structures, right? That will fill up and get bigger, and obviously during breastfeeding, lactation, release milk. And that's what this slide looks like. So this is a picture of a mammary gland during lactation. And we can see these are filled with milk. And this slide is a mammary gland. This is a picture of the umbilical cord. There's 3 blood vessels in the umbilical cord. Obviously, we'll go through and talk about that. 2 arteries and a vein. This slide is the umbilical cord. And you have a few reproductive disorders. Should be familiar, recognize them. More lecture-based, understand the specifics of the physiology. But we have cryptorchidism, which is basically undescended testicles. Obviously, if they're staying in your body cavity, they could get warmer than they should, and that can affect sperm production, etc. Hypospadia, basically your urethra is in the wrong spot, right? We can see it here coming out the bottom. Breast cancer, right? Pretty aggressive picture. Mammogram, obviously we'll talk about cancer detection and all the other stuff in lecture, but a mammogram is used to detect abnormalities in breast tissue. Ovarian tumors. Syphilis, some reproductive disorders. Syphilis is syphilis, even though we have a picture of male and female respectively. Can both get it. It's the same syphilis. And gonorrhea is another one. And we'll talk about some of these in lecture as far as causes and things like that. Testicular torsion, when the blood vessels and structures of those testicles get wrapped up, obviously could affect blood flow and could cause serious problems. Fractured penis, obviously, it's not a bone, but there are blood vessels that can be damaged and lead to bruising and things like that. And then you have your real anatomy pictures. You have them. You have homework. You're going to go through. You're going to do them. On here, they're labeled. It's great practice. I can also show you some of these pictures on your test, besides just the models and some of the questions pertaining to spermatogenesis and oogenesis. Corpus cavernosum, seminiferous tubules, all these structures we talked about, you should be able to recognize them. So, what did we talk about? We introduced male anatomy, how sperm was produced, and some histology slides. We also introduced endosperm, obviously. Female anatomy, a little bit of what's happening in the ovary and kind of development of that oocyte and release during ovulation. We also looked at some female histology, as well as some reproductive disorders. So other than that, make sure you study up, be familiar, be able to recognize models, real anatomy pictures, etc. And I'll see you in the next lab.